to thank the organizers for inviting me to participate. This is my first time attending an ICMS symposium. It's really great to connect with the community. So my lab works on KATP channels, and today my talk will focus on KATP channel pharmacology, which actually started way before the channels were even discovered. So in 1942, while studying the effects of sulfonamide compounds on typhoid fever, Marcel Show-Ling found the profound hypoglycemic effects of sulfonylureas, which led to the use of sulfonylureas to treat type 2 diabetes. And 40 years later, the first KATP channel was discovered by Akinoma in cardiac myocytes, and soon after, similar KATP channels were found in pancreatic beta cells and other tissues. And then it was then established that sulfonylureas actually inhibit KATP channels to stimulate insulin secretion, hence its hypoglycemic effects. And around the same time, another class of compounds called potassium channel openers, such as diazoxide, was found to have hyperglycemic effects by stimulating KATP channel activity. And the connection between sulfonylureas and KATP channels led Lydia and Joe Bryan to label a high affinity sulfonylurea receptor SGR1 with I-125 glibenclamide, which led to its cloning. And when combined with KRR6.2 cloned by Suzumo Seimano's group, the first KATP channel was reconstituted. We now know that KATP channels are a family of molecular sensors of cellular metabolism that regulates a wide range of physiological processes to maintain homeostasis. And the channel consists of two protein subunits, the KRR6 channel pore subunit and the regulatory sulfonylurea receptor subunit. And these two proteins are encoded by two pairs of genes in humans, and different SUR isoforms and KRR6 isoforms can co-assemble to form different subtypes of KATP channels with distinct tissue distribution, gating properties, and physiological functions, with the three main types being the pancreatic neuronal subtype formed by SGR1 KRR6.2, SGR2A KRR6.2 in cardiac tissues, and SGR2B KRR6.1 in vascular tissues. And dysfunction of these different KATP channels is linked to a number of human diseases in the endocrine neuronal tissues, for example, congenital hyperinsulinism, neonatal diabetes, and a Denz syndrome with neurological defects, as well as the cardiovascular diseases, especially the CAN2 syndrome caused by gain-of-function mutations in the vascular KATP channels. So the activity of KATP channels inside the cell is mainly governed by the intracellular ATP to ADP ratios. ATP inhibits the channel by binding to the KRR6 inhibitory site, whereas ADP in the presence of magnesium binds to the second nucleotide binding domain of the regulatory SUR subunit to promote dimerization of the two nucleotide binding domains, which then counter the inhibitory effect of ATP to stimulate channel activity. And as I already told you, sulfonylureas inhibit channel activity, such as low affinity tolbutamide and high affinity glibenclamide. There's also another class of inhibitors known as glinides, commonly used to treat type 2 diabetes. and they act by stabilizing the inhibited conformation, whereas potassium channel openers like diazoxide, phenacetyl, chromocalin, they actually stabilize the channels in the open conformation. So although there's a long history of using KATP channel modulators to treat human diseases, challenges remain due to the overlapping pharmacology of KATP channel isoforms. For example, sulfonylureas not only inhibit pancreatic KATP channels, but also cardiac and vascular KATP channels, although with about more than 10-fold less potencies. And efforts trying to use sulfonylureas, for example, to treat CAN2 syndrome caused by gain-of-function KATP mutations in the vascular KATP are hindered by the fact that these drugs cause profound hypoglycemic effects by acting on the beta cell KATP channels. And diazoxide, which is the mainstay treatment for the genetic disorder congenital hyperinsulinism, has a lot of undesired side effects as well. For example, excessive hair growth. Diazoxide basically acts like minoxidil, which is the main ingredient in Rogaine, to stimulate hair growth, and it can also induce cardiovascular defects resembling CAN2 syndrome. So for these reasons, there's been renewed interest and effort in trying to identify new KATP channel modulators. So for example, Jared Denton's group at Vanderbilt has done a lot of high throughput screen of large compound libraries using a thallium flux-based fluorescence assay, where he has reported a SGR1 selective activator as well as SGR2 selective inhibitor. And my group has tested compounds that have been shown to modulate other ABC proteins, specifically CFTR. So based on David Thomas's group at McGill, showing that carbamazepine actually can correct the CFTR delta F508 trafficking mutation. And because CFTR shares ABC core structure with SGR1, so we reasoned that these compounds might also act on KATP channels. So indeed, we found that carbamazepine, which is actually best known for as an anticonvulsant by inhibiting voltage-gated sodium channel, is a really potent KATP inhibitor with KD around 20 nanomoles, but it also can rescue trafficking mutations in KATP channel, which I'll come back to later. The third approach is to test compounds that are derivatives of known KATP modulators. And this is an effort in collaboration with Rhythm Pharmaceuticals, and it's a result of a mother who has a son with congenital hyperinsulinism and who was really frustrated with the side effects of diazoxide. So this project mainly focuses on trying to find SGR1 selective openers, and this is work unpublished, and I just want to show you that using a non-radioactive rubidium efflux assay, the ion channel reader from Aurora, that we were able to find many compounds that are more potent than diazoxide at the same 10 micromolar concentration tested. And although I'm not showing you here, these drugs don't seem to activate the SGR2 containing KATP channels. And finally, we use a structure-based virtual screen of compound libraries to try to identify novel modulators. And in the remaining time, I will focus on our recent work that led to the identification of a compound that is a KATP inhibitor chaperone. So first, just a little bit background about KATP channel assembly and trafficking. So surface expression of KATP channels require co-assembly of these two structurally very different proteins into an octamer in the ER. Unassembled or misassembled subunits are retained inside the ER, unable to exit the ER to the cell surface. And this is due to arginine-based RKR retention signal found in both subunits. This was discovered here at UCSF by Lily Jan's lab. As you can see in this Western blot, that normally SUR1 after trafficking past the Golgi becomes complex glycosylated. However, in the absence of its assembly partner, you don't get this upper band, and the immature SUR1 band is retained inside the ER. And it turns out that a lot of the loss of function mutations identified in congenital hyperinsulinism actually cause misassembly and trafficking of the channel to the cell surface. Here I have mapped a lot of the trafficking mutations on the structure. You can see that a lot of them are clustered in this TMD0 domain, the N-terminal transmembrane domain of SUR1, which makes direct contact with the KR6.2. So it's a primary anchoring domain for channel assembly. So early on, my first postdoc discovered that if we incubate the cells in globenculamide, which is a high affinity sulfonylurea, we can actually rescue some of the trafficking mutants to the cell surface. Here, as an example, the A116P in SUR1, you can see the appearance of the complex glycosylated band after treatment, and also in immunofluorescence experiments, you can see the recovery of surface KATP channel. So to understand how the drug can exert such effects, my former student, Greg Martin, solved the cryo-EM structure of KATP channels bound to ATP and globenculamide, and he was able to find the globenculamide density in the transmembrane pocket in the first the transmembrane helix bundle above the NBD1, first nucleotide binding domain. And here's a closer look at the binding site here, and this is top-down view. And we also determined the structure of SUR2B KR6.1 channel. This is a vascular channel bound to globenculamide. And you can see the slightly different poses of this compound in the two different SUR isoforms. And I want to bring your attention to these two amino acids that are different between the two isoforms, which likely play a role in determining the affinity of these KATP of this compound to different KATP channels. So we then went ahead and determined multiple structures of the channel bound to different pharmacochaperones that we had characterized at that time, and we were able to show that they indeed all bind to the same binding pocket, so suggesting that these compounds act through a similar mechanism. But we still didn't understand how inhibitor binding at this ABC core domain could inhibit channel activity and rescue the trafficking defects caused by mutations in another transmembrane domain. And the clue came from our structural function studies showing that the KRR6.2 N-terminal plays a critical role in mediating the effect of these inhibitor chaperones. As you can see here, deletion of the N-terminal 30 amino acids abolish the ability of glibenclamide and carbamazepine to rescue this SUR1 F27S trafficking mutation, whereas deleting the C-terminal 25 amino acids had no effect. And when we looked at our structure more carefully and zooming it out at lower resolution, we were able to see this density corresponding to the N-terminal of Kr6.2 that is wedged between in the cleft formed between the two transmembrane bundles of the SUR1 ABC core, right next to the bound drug. In this case, it's repaglinide, a glenide drug. So we used an engineered cysteine pair to do cross-linking experiments and showed that cross-linking under oxidizing condition induced spontaneous channel inhibition in the absence of any inhibitors. And this could be reversed by adding reducing reagent to break the sulfide bond. So these experiments really showed us that the KRR6.2 N-terminal play a critical role in mediating the chaperoning and inhibitory effects of these drugs. So this is just a cartoon showing, summarizing what we found. We think that during channel assembly, this N-terminal of KR6.2 formed transient interaction with SUR1 to allow the TMD0, which is the primary assembly domain with KRR6.2, to form an octameric complex. This is consistent with the observation that if we delete the N-terminus, we also dramatically reduce the efficiency of biogenesis of wild-type channel. And for the mutant, we think that the mutations probably cause suboptimal conformation of the domain to prevent assembly with KR6.2, but when you have the drug bound here, it really stabilizes, it traps the KNTP or N-terminal of KR6.2 in this interface to allow this mutant TMD0 domain to form complex with KR6.2 to traffic to the plasma membrane. So as you can see here, then that the inhibitory effect of these compounds and chaperoning effects go hand in hand. But the compounds we have discovered so far, they are very high affinity. They are irreversible, so we cannot wash out the compounds to recover channel function. And there are also side effects of other compounds such as carbamazepine and low affinity sulfonylurea. So this prompted us to conduct a structure-based virtual screen of new KATP for new KATP inhibitors. And this was in collaboration with the company Atomwise through their academic collaboration program. And using a repaglinide bound structure as a template, we screened the compound, the anamine compound library. And then my former student, Asma, carried out experimental testing of the top storing compounds. And as you can see here, this compound that we chose to characterize further, which she named a capron, can rescue the F27S mutant here on Western blot, but also other trafficking mutations I'm not showing here. And it can inhibit channel activity, but also in a very highly reversible manner. And it has a higher affinity for the SUR1 KRR6.2 channels than the SUR2 containing cardiac and vascular channel. And using rubidium efflux assay, we showed that we can recover channel function in intact cells after overnight treatment, followed by a quick wash. So my postdoc then determined the structure to confirm that this compound indeed binds the pocket that we used for the virtual screen. And we conducted mutagenesis studies to validate the binding site model. Interestingly, using MD simulations, we found that although most of the molecules form stable interaction with the channel protein residues, this imidazole group is actually highly mobile. Whether this explains the lower affinity of this compound needs to be studied, but it's an interesting observation. So this is just a quick comparison of all the inhibitor chaperones that have been reported today. And you can see that really this is quite a promiscuous binding pocket that can accommodate really diverse set of compounds. So we think there's probably still room to exploit this pocket to really find compounds that is tailor-made for your purpose. So right now we are collaborating with Shyng in Academia Sinica, who is in the audience right here, to actually see whether these compounds can be used to restore insulin secretion and blood glucose regulation in a mouse model. So you can see here, these are data generated by Shyng's lab. These mice show transient neonatal hypoglycemia without effects on body weight. And these mice, when they are a bit older, 2 to 3 months, they show glucose intolerance without much change in insulin sensitivity. And these are phenotypes that are very similar to SCR1 or KR6.2 knockout mice, suggesting that there's no KATP channels here. Indeed, using whole cell recording, they were able to show that where heterozygous mutant have robust KATP currents that are stimulated by diazoxide and inhibited by glibenclamide, in the homozygous mutant mice, there's no evidence of KATP currents. So now we're ready to use these mice to test the effects of the inhibitor chaperones that I told you about. So just to summarize, this is a cartoon. What I think is that the cryo-EM structures really now allowed us to understand the mechanisms of how these inhibitors and openers work. Openers structures have been determined mostly by Lei Chen's group in China, which I don't have time to talk about. But in any case, I think that a better understanding of the mechanism of drug actions will lead us to a better KATP pharmacology for KATP channelopathies. So with that, I would like to thank all the people who contributed to this work. Right now, I have a very small lab, and these are past members who contribute to the work that I highlighted today. And these are my collaborators. They're wonderful. Ha was involved in virtual screening from Atomwise, and Kasha was responsible for MD simulation, and Shyng is helping us with the transgenic mouse, with the knocking mouse study. And I want to acknowledge the support, cryo-EM support from OHSU and PNCC, and the funding. Thank you.
Thank you for a great presentation. We have some time for questions. And maybe to start out the discussion, so I was just wondering about the virtual screen and what sort of the conversion rate then to your screen was. I mean, how many of the virtual compounds that you identified were then sort of shown also to have a hit also in a functional assay?
Yeah, thank you for that question. So the Anamine compound library has about 2.5 million compounds, I think. So we chose it because they are commercially available, so we could test them experimentally very easily. And we tested about the top. The company was actually very nice. They sent us the compounds for testing. So we tested about 100 of them. I would say about 10% of them showed some effects in the Western blot screening for chaperoning activity, and then followed by electrophysiology. So it was a bit of a painful process. initially we had meant to use rubidium efflux, radioactive rubidium efflux to do the inhibition screening, but the radioactive rubidium was discontinued, so we were scrambling trying to find an alternative assay. So we ended up with the rubidium efflux assay to help screening, because I was not able to screen all the compounds manually by patch clamp recording.
Fantastic talk. Two quick questions. How specific is this compound A-capitronis for 6.1? Does it have any effect on the 6.1 in addition to 6.2? And a second question, can either of these channels be also regulated by cholesterol or cholesterol derivatives?
Yeah, I think those are good questions, and the compound does act on SGR2 containing channel, and SGR2A or 2B, basically the binding site is exactly the same for these two SGR2 isoforms. So the affinity is about 5-fold difference. Yeah, so it is SGR1 selective, but I think obviously the selectivity needs to be improved, and so medicinal chemistry follow-up studies would definitely be very important. The other question about other regulators, I didn't have time to talk about this, but obviously KATP channels are also regulated by PIP2, and so in the process of determining channel structures bound to PIP2, we also observed other lipid molecules, including things that we suspect as cholesterol, but we have not followed up on it. But they definitely decorate the complex in various places, especially at interfaces of the two subunits. Yeah.
I guess I was curious how similar the binding site is to like the corrector CFTR.
Yeah, I knew somebody was going to ask this question. Yeah, so it's actually quite interesting. The type 1 corrector is binding kind of more close to the membrane cytoplasm interface, a hydrophobic pocket. So it's quite different from what I described to you. But the type 3 corrector, so there was a paper from Zhu Chen's lab that looked at the trifecta, or basically three combination of two correctors and one potentiator, where they identified the binding sites for all these compounds. And the type 3 corrector actually looks very similar to the binding site that I described. And remember, I told you that carbamazepine binds to this pocket and has been shown to have corrector effects on CFTR delta F508 also. We have not looked at that directly, and Zhu Chen told me that she tried to get her students to work on it, but nobody seemed to be interested. So to see whether KATP correctors also bind. But we know that glibenclamide also inhibits CFTR to some extent, low affinity, yeah.